Hello, my name is Bruno. I'm doing a presentation about cloning. Hope you guys enjoy. Thank you. Let's talk about cloning. What is cloning? Cloning are organisms that are exactly genetic copies. Every single bit of the DNA is identical. Cloning can happen naturally, like identical twins are just one of many examples, or they can be made in the lab. The term cloning describes a number of different processes that can be used to produce genetically identical copies of biology entities. It's a very complex process. And to this day, scientists are still trying to figure out how to make clones.
As cloning is done? Many people first heard of cloning when Dolly the sheep showed up on the scene in 1997. Artificial cloning have been around much longer than Dolly.
There are two ways to make an exact genetic copy of an organism in the lab: artificial embryo and twinning somatic cell nuclear transfer. Artificial embryo twinning. Artificial embryo twinning is relatively low-tech way to make clones. As the name suggests, this technique mimics the natural process to create identical twins.
In nature, twins form very early in development when the embryo splits in two. Twinning happens in the first days after egg and sperm join, while the embryo is made of just a small number of unspecialized cells. Each half of the embryo continues dividing on its own, developing into separate complete individuals.
Artificial embryo twinning uses the same approach, but it carries out the petri inside of the mother. The very early embryos separate into individual cells, which allows to divide and develop for a shorter time into the petri dish. The embryos are then placed into the mother, where they finish in development. Again, since all the embryos came from the same fertilized egg, they are genetically identical.
Option 2, somatic cell nuclear transfer, uses a different approach than artificial embryo twinning, but produces the same result, an exact genetic copy, or clone of an individual. This was the method used to create Dolly the sheep.
We have Professor Keith Campbell with us, the creator of Dolly the sheep. He will explain to us a little bit about cloning. So Doc, tell us a little bit about cloning. What's cloning for you?
Yes, Doc, tells us a little about cloning.
Cloning has numerous applications, both in applied and basic science, in medical applications, in agricultural applications, potentially in preservation of species. In terms of agriculture, which is not the major use that cloning will be put to, it allows us to preserve genetics, so we can actually store phenotypes, so we can store both males and females. We don't just store germ cells. But it also allows us to multiply up elite animals so that we can disseminate the good quality genetics or the traits we want into the population as a whole much faster. Because you have to remember that agriculture and breeding is now worldwide. It's not just the farmer down the road where you swap bulls. Now the genetics are being shipped all over the world. And so by multiplying up, say for instance a very good quality sire, you can ship out loads of semen to many places around the world. Now that isn't going to shrink the gene pool, it's actually just disseminating good traits. But the other thing, because we can actually preserve genetics very simply, we can at a later date go back to those. This is also relevant to rare breeds of farm animals who may have traits which we have not identified yet. They may be more resistant to disease, which we may want to breed into the population in the future.
We have here with us another special guest. His name is Dr. Mike Lepov. Hi, Doctor.
Hi, Doctor. Thank you for coming on the interview. Can you explain to us where you are working now and how is it related to cloning?
So we in our laboratory, we interested in developing a new way of creating stem cells, but in this case the stem cells would be from a cell or skin cell derived from a patient. So that way the stem cells we create, they would be compatible if we transplant these tissues back into patients. So these stem cells are kind of very early unprogrammed cells, but they have of course capacity to become any other cell type. And so the task is now to make these different cells, for example neurons or hepatocytes or blood cells, whatever the patients need, to produce in a petri dish, basically in the laboratory, and then transplant these tissues and cells back into patient, already mature these cells, so that we could treat some of the diseases caused by lack or dysfunction of these cells in the body. So widely used example is Parkinson. So the Parkinson model, Parkinson disease is caused by dysfunction of very few types of cells. So basically it's one type of neurons that not producing enough some chemicals. And in many patients these cells are actually gone, they died off. And there are no other cell types that could make this chemical. So now in the laboratory we can learn how to produce, for example, these neurons that would produce this the chemical and transplant this into patients with this disease.
I have Dr. Rob Lenzer with us. Dr. Rob Lenzer, I would like for you to explain what you're working now. If you could please give us a short explanation, a little bit about cloning and what exactly we're working now. Thank you.
So I'd like to speak to you about the use of cloning in stem cells to resurrect life. And as you know, there are two ways to make copies of cells and organisms. The first and most controversial, of course, is cloning, and that is also known as somatic cell nuclear transfer. And the concept there is very simple. You start out with basically an empty egg, that's the large circle here you see there, and then you place the cell you want to clone, the smaller somatic cell right next to it, and then you send an electrical charge through the unit, and what that does is it damages the membrane between the two, and the nucleus of the cell you want to clone dumps into that empty egg. And then what you do is you add some chemicals, you fool that unit into thinking it's fertilized, it starts to divide, and you end up with what's known as a pre-implantation embryo. And then you can do one of two things with that. You can place that in a petri dish where you can turn that into embryonic stem cells, which are the master cells of the body, and they can turn into virtually every cell type. And then the other alternative is that you can place that into a surrogate animal to create an entire organism. Another approach that's newer is known as cell reprogramming, and that leads to what's known as induced pluripotent stem cells, or iPS cells. And what you do is you start out with a somatic cell or a piece of skin, you throw in some transcription factors and bring that differentiated cell back to a state of pluripotency, very much like an embryonic stem cell. And we have new tricks now where we can actually turn those cells into an entire organism as well. So to date, about 24 different species have been cloned. Back in 1958, John Gordon actually cloned the first animal. That was a frog. In fact, he was just recognized for that feat a few months ago with the Nobel Prize. And since that time, of course, has been Dolly the cloned sheep, and we and other groups have cloned mice and goats and even cats and dogs. In fact, back in the 1990s, we actually cloned an entire herd of cows from genetically modified cells. So what you actually see here are animals that are making human albumin in their milk. So we could use that same approach to reconstruct extinct animals a la Jurassic Park. So in this case, we took a skin biopsy from the ear of a cow, we grew up the cells, knocked in a gene cassette, and then used ordinary eggs to create that herd of animals. So similarly, as George Churchill described, we can then take, say, an elephant cell, knock in the genes for tusks or long hair or even hemoglobin so it can live in a cold climate, and then using a technique that I'll describe later, we can then create sperm and eggs and indeed an entire organism from that. So there are two types of cloning. One is known as interspecies cloning, and the other is intraspecies. So with the intraspecies, you actually use the egg and the cell from the same species you want to clone, but using this cross-species approach, we actually can take the egg of one species to clone the cell from another. And that's very important if you want to resurrect extinct animals or if you want to clone endangered animals. So back in 2000, we actually used this cross-species technique to clone the first endangered species. In this case, it was Agar, which is a wild ox on the verge of extinction. And at the time, everyone said that, that's not going to work, that's impossible. And the reason for that is that a clone isn't really entirely a clone. It turns out that every cell has two genomes. One is the mitochondrial genome, and the mitochondria are the organelles in the cell that make energy. And that's maternally inherited, so that will come from the egg. And then the other genome is the nuclear genome, and that's what contains the genes that distinguish you and I from an elephant or a mouse. So those two genomes have to talk to one another, and there's evidence that that can only occur within 8 to 18 million years species radiation. And so we got around that problem by actually using very closely related species, concordant xenograft combinations. So in this particular case, we had a gaur and a cow.
Like any other great innovative idea, cloning is no less than groundbreaking idea. It's a very exciting research subject with plenty of prospects. But the reason behind cloning should be justified for research to be carried forward. Any scientific research nowadays should be purchased. Keep in mind there should be benefit for the society and less hindrance or problem. That said, I believe the therapeutic cloning should be encouraged as dealt with regrowth manufacturing organs as a result of life-threatening disease. I think the decision about whether it would be ethical to clone the organisms, body parts, etc., would have to be based on around degree of self-awareness.
Human cloning is a very controversial subject. It can disrupt the balance in the society, which is having unique identities of everybody, adding to the fact that it is a great hindrance of natural process that has always existed on this planet. Cloning can lead to wars, misunderstandings which will not be positive.
Cloning can be very beneficial, saving lives, help curing disease, understand the genetics a little bit better, help animals, end the hunger in the world. But we gotta be very careful, because it was as good as it's also bad. It can lead to wars and extinction of the human race. It can lead to a lot of several things. We need to be very careful. I have no problem at all or any issue with cloning. Cloning also can lead us to live a longer and healthier life. Moral of the story, we just gotta be very careful, and people need to be self-conscious about this.
Cloning can revolutionize the world and the way we live, or may be so minimal that it would not affect us at all if is allowed. I think each person must decide for himself, or herself, if they believe that cloning should be allowed or if the government should intervene it. As fascinating as it is, we need to approach this subject with carefulness. Thank you very much for listening to us. I appreciate everyone's time. Until next time.
Alright guys, it was very nice doing this documentary and presentation. I had a lot of fun doing it. Now I'll leave you guys with a very provocative video about human cloning by Robert London and Morgan Freeman through the wormhole. Thank you very much, and you till next time. Bye.
Imagine a world where a woman could bring back her deceased husband by giving birth to him. Or a man could bring back his mother and raise her as his daughter.
If we have a living cell of a dead person, certainly we could in theory clone that individual. We published a paper about a year or two ago where we showed we can actually now create human embryos that are genetically identical to a normal embryo. The only way you would really know whether you could clone a human being is to actually implant that embryo into the uterus of a surrogate mother. Of course, we cannot implant those. That would be considered unethical, so it's unclear whether or not they would give rise to a human being.
Society has vehemently rejected reproductive human cloning. In this climate, it is extremely difficult for geneticists to obtain funding for their research. There is also a shortage of material.
So one of the problems with human cloning is the supply of eggs. So with a mouse or a cow, we can get literally hundreds, if not thousands of eggs. We can go to the slaughterhouse, for instance, and get them for $1 each and get thousands of these cow eggs. With the humans, it took us over a year just to get 5 eggs.
Even if you had the eggs, it could take hundreds of pregnancies to perfect human cloning, a process that could result in scores of babies with horrific genetic damage.
It'd be very much like if you wanted to clone your child, it'd be like sending them up in a rocket with a 50-50 chance it would blow up.
In most of the world, cloning humans is not illegal. One day, a rogue scientist will pull it off. But cloning a person is not the same as duplicating a person.
A lot of people, who have their pets, they want to often clone them, and they want Fluffy back. And what I tell them is, you're not going to get Fluffy back. As a matter of fact, we actually cloned entire herds of cows from a single cell from the same animal, and they develop a whole hierarchy, just like we do in humans. So you have timid cows and aggressive cows, and they're all clones. So they develop their own behavioral patterns. So the environment has a very profound impact on your development.
Let's say we decide to take DNA from Einstein's hair and grow some new Einsteins. Those clones would not be the man who wrote E = mc squared. Each would have a unique personality shaped by his environment. Clones are like identical twins born years apart. They may be similar, but they will not be the same. Perhaps the key to life after death is not to grow an entirely new body, but to resurrect the one you have.